Hello, today we're going to talk in a little bit more detail about different types of environments or solutions and the effect they can have on different cells. So, either animal cells or plant cells in our examples today. If you recall, osmosis is the diffusion or movement of water down a concentration gradient or with a concentration gradient from an area of high concentration to an area of low concentration. This process requires no energy. That being said, we're going to start by talking about isotonic solutions or isotonic environments and how that can affect a cell and the directions that water can move or not move. In this first example, within an isotonic solution or environment, we have to note, and I've kind of drawn it in blue here, the little blue dots represent water. And there's an equal amount of water. I tried to draw that as best I could in terms of the little circles inside and outside this animal cell. If we look at the plant cell, we have that same situation. But you may notice the plant and the animal cell feel a little bit different about the environment. In an isotonic environment or isotonic solution, a plant cell is actually a little bit flaccid or limp. Plant cells aren't unhappy, but they're not like happy. An animal cell, on the other hand, is very happy in an isotonic solution or environment. Again, this means there's an equal amount of water inside and outside of the cell, and it's not going to cause any changes in the cell. As we move forward and we take a look at hypertonic solutions or environments, you may notice that I made this E like extra kind of like skinny. I like stretched it out there. And I did that to hopefully get you to make a connection that in a hypertonic environment or solution, water is going to move out of the cell. And that's because the environment doesn't have a lot of water in it. So there's actually more water inside of the cell than outside of the cell compared to the outside environment. So water moves out and the cell shrinks. So the animal cell here gets shriveled and is unhappy. The plant cell here gets plasmolized. That's a fancy science word for the cell membrane pulls away from the cell wall. So the cell wall is outside here, and then this is the cell membrane on the inside in black, and it pulls away from the cell wall, and the plant is unhappy as well. You may also notice that the central vacuole is way shrunken down. That large central vacuole is really responsible for holding water in a plant. Lastly, here in terms of environments, we have a hypotonic solution or environment. You'll notice I made this O in hypotonic kind of oversized, and that's because water is moving in to a cell when it is placed in a hypotonic solution or environment. When the water goes into a particular cell, if it's a plant cell, the plant cell is actually really happy. That large central vacuole is full of water, and the scientific name for when the plant has a lot of water in its cells is turgid. It's firm. This would be a plant that its leaves are perky and happy. On the other hand, with an animal cell, if too much water goes into the cell, it can actually break or burst. And that's what I'm trying to represent here, almost like a balloon that's broken. The fancy word for that is the cell has lysed or has gone through lysis. Um, and that would really be bad if you are an organism that has animal cells and your cells burst. So, again, the environment has essentially too much water. The water rushes in. And in the case of an animal cell, that is dangerous. And that's my little summary of the different types of solutions and environments. And I hope that was helpful. Thank you.